Okay, so good morning, everyone. Welcome to the second series of webinars that are hosted within the Cedric project. So my name is Yuri P. I'll be the host of today's seminar. And before we start with the actual seminar, I would like to introduce you to the Cedric project. So Cedric, Cedric is a project which has been financed within the Interreg Italy Austria initiative. And the title of the project is Creation of a Sustainable Cross-Border Agroecosystem by Leveraging Root Microbiome Biodiversity. And the idea of the Cedric project is to actually strengthen the microbiological diversity in areas characterized by soil which are poor or with an altered microbial ecosystem. Within the Cedric project, we have several units participating. There is the ECGB of Trieste, which is the coordinator. Then we have, of course, my university, the Free University of Bolzano, the University of Innsbruck, and then we have several other participants like SICIT Group, Eichenbichler, Biotreat, and also ERSA. You can find all the information about the project following the link that you have here or scanning the QR code. So the main objective of Cedric is to develop an efficient method to transplant microbiome. So from healthy rhizosphere to roots, and also to create a platform in which we aim at promoting the translation of the scientific knowledge into sustainable method to be applied at field level, and also to develop a training program which is focused on the role of healthy microbiome in soil ecosystem and the benefits associated with it. And this is actually the aim of today's webinar. So this is, as I mentioned at the beginning, the second series, because we ran a first series of webinar last year, which was quite successful, and we were asked to organize a second series. And so here you have the first reappointment of this initiative. Today, we will have a seminar presented by Professor Vito Armando Laudicina from the University of Palermo. Before leaving the floor to Professor Laudicina, I would like to introduce him first because he's an esteemed colleague, but also because he's a dear friend of mine. And Professor Laudicina basically got his PhD in pedology at the University of Palermo, and then he moved to Spain for a postdoctoral fellowship on soil carbonates in semiarid environments, followed by an OECD fellowship at the Scottish Crop Research Institute in Dundee. Now, at the moment, Professor Laudicina is full professor of agricultural chemistry at the University of Palermo, and his team's ambition is to translate soil quality and carbon research into sustainable practices, including wastewater reuse and biodegradable mulching solution. So before leaving the floor to Professor Laudicina, I would recommend all the participants to make sure that your microphones are muted during the presentation in order to avoid any background noise. We will have a question and answer session at the end of the webinar, but in any case, during the webinar, feel free to write any question in the chat, and then we will take care at the end during the QA session. And the webinar is going to be recorded, and the recording will be shared on the project website. So I would like to give the floor to, at the moment, I would like to give the floor to Armando. for his presentation. So I'm stopping the sharing on the screen and I invite him to share his. Please, Armando.
So again, many
Great.
thanks to Professor Yuri from Yuri P from the University of Bolzan, and again, good morning to everybody. Many thanks for inviting me to give this webinar. It is very nice to present the results of the research that I've carried out here in Palermo with some collaboration from with other university in Italy, but also from Germany and Spain and so on. So the title of the webinar is Innovative Mulching Films for Sustainable Agriculture. It is a very important and hot topic during the last years, the theme of plastic. So I think that all of us know what is plastic, very common materials that we find in all the objects that we found around us. Plastic is a material that is composed by a polymer. Generally, it is a polyethylene that can be easily shaped, and it is valued for its versatility and low cost. But just due to its durability and polymer structure leads to serious environmental issues. Just to give some numbers about how much common is this material around us, it is estimated in 2021 that about 391 million of tons of plastic was used just in 1 year. Just in 1 year. And of all this plastic, as you can see here, about 90% is coming from fossil-based sources, and only a very small amount of this is coming from bio-based sources. And another important aspect is that regarding the amount of this material that can be recycled, only 80% can be recycled. And just to go in deep with this number, in this other picture that we have here on the right part of the screen, as you can see, only 4% of these 390 million tons of plastic is used in agriculture. So this number could appear very small, a very little amount of plastic that are used in agriculture. But if you go more in deep with this number, we can see that the amount of plastic used in agriculture, it is estimated in about 60 million of tons. That is a lot amount of plastics. And of these, about 20% is used as mulch films. So what are mulching films? Mulching films are a very teeny sheet of plastic that is used to cover generally the soil. And it is used because I have many advantages from many point of view. The most important advantages is that mulching films allow to suppress weed, so we can reduce the use of herbicide. For example, we can reduce the tillage impact on soil, and also mulching films may help to retain moisture in soil, can regulate soil temperature, can regulate also erosion in soil, in sloping soil, and generally it can improve crop yield. We conducted also a research about the impact of plastic, for example, in in an orchard, and comparing this mulching with long-term mulching with a long-term rotary tillage. For example, we, as you can see here, we used different soil management, and what we have found it was that total organic carbon improved in soil where in soil mulched soil, but also microbial biomass. We experienced also a shift in the fungi bacteria with an increase of fungi. This was a very nice result and also expected because the soil was not tilled and so on. So we got a lot of interesting results. Also crop yield was improved because the soil, the mulched soil became more fertile. So mulch, this agrotechnique is very different advantages, but also this technique have some environmental and economic drawbacks. For example, there is there is a lot of soil that are contaminated by plastics. About 40% of European agricultural soil are contaminated, and then we can find these plastics also in other compartments. Plastics contributes also to soil degradation, reduce biodiversity. There are some studies that found a fertility decrease of about 20%. It is very expensive for the farmers to dispose of the plastic films at the end of the cropping cycle. And this is also a very important issue because plastics that are disposed of cannot be recycled because generally these films are very, very dirty. The soil is attached to these films, so the films need to be clean before they are recycled. So there are very, very important drawbacks. So the solution, or anyway, the solution, or what we think during a project that we carried out during the last years was what kind of plastics we need to use. This because, as you can see before, we need to use mulching films because they have also some advantages. So the plastic films can be classified in different classes. We can distinguish bio-based and fossil-based mulching films, but we also can classify these films in not biodegradable and biodegradable. So if you look to this picture, I think that all of you agree with me that we need to use bio-based mulching films that are also biodegradable. So this was the topic of, as I said before, my research during the last year, and in particular, thanks to a national project funded by the Italian Minister of Research, we aimed to develop a fully biodegradable and bio-based mulching films prepared by using some natural sources, some component like cellulose, chitosan, and sodium alginate. But another important aspect of our project, of our research, was that to enrich these films that are bio-based and we tested if they were also biodegradable, was to enrich these films with inorganic nitrogen and phosphorus salts. This because if really our mulching films are bio-based and also biodegradable, at the end of the crop cycle, they can be buried into the soil and may serve also as fertilizers. And another important aspect was that by enriching them with nitrogen and phosphorus, we modify the carbon to nitrogen ratio and the carbon to phosphorus ratio just to make these ratios more comfortable for soil microorganisms. So soil microorganisms can decompose these films very easily. So going to this project or the research of this project, the first step was to develop these films. We collaborate with other colleagues from the University of Palermo, from the group of physical and chemical materials, and we started to prepare some different solution that then we mixed. So we prepared the solution, a suspension with carboxymethyl cellulose, with chitosan, and with sodium alginate. As you can see here, this can be, for example, cellulose can come from biomass, chitosan from some crustaceous, sodium alginate from algae, and so on. So we prepared different suspension dispersion, then we mixed them, and we used different mass ratio of cellulose, chitosan, and other materials that I will show you then. And we used the technique of solvent casting. We prepared these films in laboratory, so we used these very nice techniques that consist of to put this dispersion in a capsule, in a Petri capsule, and allow to the solvent to be removed at room temperature. We used also an oven at 35, 40 degrees, more or less. Then after we removed, after that the solvent was removed, we get our films that I will show you later, these nice films. So first, before to use this, as I said before, we enriched these films with the inorganic salts. So very easily we take the films, and we soaked these films in a solution containing ammonium phosphate. We left the films for 5-10 minutes. We tried different times. Then we soaked also this with the calcium chloride to facilitate yes, the link between the different component, and then we get our films that you can see here on the right part of the screen. As I said before, we use the different ratio, and we tested these different films. We tested these. You can see here on the right side of the screen our films, how was easily to use, and very nice films. So we tested these films for different parameters. The first things that we did was to set up an experiment to evaluate the release of phosphorus and ammonium in solution. And what we have found was that the release was very, very fast. And this was not a nice feedback that we get, because we hope that the release of ammonium and phosphorus was as I can say, it was gradually, no, just gradual to allow also the crop to take this nutrient. But the first attempt that we did was not so what was not so nice because, as I said, ammonium and phosphorus was released very, very fast. So what we did after these results was to change our experiment, our method of preparing these films, and we moved the supply with the addition of nitrogen and phosphorus. We moved this, sorry, here. What we did was to add the solution with containing nitrogen and phosphorus directly in the dispersion. So this salt can be part of the lattice of our films. So after this change, we got a very nice results. We slow the release of ammonium and phosphorus, and also we characterize these films studying their mechanical properties, and also you can see here the elemental composition, and we get a film that was very, very near to the films that are at this moment commercially available. Also with regard to the elastic modules, the elongation at the brick, and so on. So these films, in particular, the films where we used a ratio 1:1 between cellulose and chitosan, we get a very nice results with these films. So then what we did was to assess if our films are really biodegradable. We are sure that our films are bio-based, but are they really biodegradable? So we did another experiment according to the method described in the ISO 17556, and according to this method, at least the 90% of the plastic that we added to our soil have to be degradable. So what we did, we selected the soil according again this method. We added our plastic films and we incubated these films in laboratory. And so what we got was that our films, as you can see here, the yellow one and the red one, release about or more than 90% of the carbon that we had into the soil after 120 days. So according to these results that are already published we can say that our films are bio-based and biodegradable. So the next step was to evaluate these films, to evaluate the effect of these films on soil properties, and in particular on the dynamics of nitrogen, of phosphorus, and on soil microbial community. So we did again another experiment where we had these films to the soil. We used sandy soils because generally mulching films are used in horticulture, and the soil that at least in Sicily or in the Mediterranean region are used for horticulture are sandy soils. So we select a sandy soil with the neutral pH, with a good content of organic carbon. We incubate this soil with our films, And what we have found was that, for example, with regard to the nitrogen dynamics, during the incubation, as you can see here, the amount of ammonium decreased and the amount of nitrate increased. This was a very nice result because this suggested that during this incubation that was of 120 days, the amount of nitrogen available for plants can increase. And our films stimulated the nitrification process. Another important aspect was that about the available phosphorus. And also, as you can see here, we got a maximum amount of available phosphorus that slowly increased during the incubation. We got the maximum amount of available phosphorus after 60 days. So this is also another nice results. And also soil microbial community increased after 30 days. As you can see here, we got a very nice increment about increase about carbon microbial biomass and also about nitrogen in the microbial biomass. So these results are very important for us. And we investigated also what happened to the microbial community. And what we got was that the fungi bacteria ratio did not change. It was we observed a slight decrease, but it was not significant different. But we experienced an increment of the gram-positive bacteria. That is a nice feedback for our microbial community. So another important aspect that we investigated was that about the microbial activity. We investigated the different enzymatic activities, and what we found was that it was stimulated. So we improved the nitrogen and the phosphorus availability. We improved the microbial biomass, and also we stimulated with these films the microbial activity. Indeed, as what we have found was this a nice increment about beta-glucosidase at after 30 days. Beta-glucosidase is very important because it contributes to the to broken the bonds between the molecules that compose our films, both in the carboxymethyl cellulose, but also in the chitosan. We have glycosidic bond, but we investigated also, for example, nitrogen acetyl beta glucosaminidase that contributes also to broken other bonds that we imagine we can have in our films, and also, for example, the lipase activity that contributes also to the breakdown of our films. So this suggested us that, as I said before, that our films stimulated microbial biomass, stimulated microbial activity, and for this we experienced an increase of nitrogen availability and phosphorus availability. Then to go more in deep with our results, to understand better how our films works in soil, we performed another experiment by using carbon-13. These techniques is very useful to trace the fate of carbon that we add to the soil. So we prepare our films enriched with carbon-13. It was very expensive. So we prepared our films with the carbon-13, and we incubated our films into the soil. This experiment was performed at the University of Hohenheim in Germany with the contribution of the team of Professor Ellen Kandler at the University of Hohenheim. And so we used two different soil with a different level of fertility, a soil, a silty clay soil rich in organic carbon, and another soil that was not so rich in organic carbon. This because why we used the carbon-13, because as I said before, carbon-13 allowed us to understand the fate of carbon. What I mean is if this carbon that we had by using bio-based and biodegradable mulching films is mineralized by soil microorganisms or are immobilized by soil organisms, and in which percentage. So we performed this different experiment. We analyzed the different parameters during this experiment. For example, the accumulated and hourly emission of carbon dioxide, the release of ammonium nitrate and available phosphorus, microbial biomass, enzyme activities again, and as I said before, the isotopes analysis. So what we observed was that, as you can see here in the first two graphs, that there was an increase of the carbon dioxide released by the two soil, but with a different percentage. In the less fertile soil, the increase of carbon dioxide was very, very high compared to the fertile soil. That means the soil rich in inorganic carbon. And we observed also that also the rate of carbon dioxide was the highest in the less fertile soil. So this is what means. This means that the soil that are limited in nutrients and in particular in carbon respond strongly or stronger than the fertile soil. Other parameters that we investigated, as I said before, was ammonium nitrate, available phosphorus, and we found the same results that I showed you before. So this confirmed the nice impact that have our films in the soil. And another important aspect, another important results that we have found was that this carbon that we added by using our films, this carbon is immobilized by the microbial biomass. A good amount of carbon that we had is immobilized by our microbial biomass, the soil microbial biomass. Why this is a very important aspect? Because soil microbial biomass, soil microbial biomass, soil microbial community is the engine of our soils that allow that many ecosystem service can be done by our soil. So by adding these mulching films, we stimulated again microbial biomass, and the amount of carbon that we added to the soil in part was respired, but also in part was immobilized by microbial biomass, thus suggesting that microbial biomass increase. And the other important aspect that you can see here, the different blue line and the red line, was that by adding the nitrogen and the phosphorus in our films, as you can see here and here also in the right, on the right part of the screen, we got a greater amount of carbon that can be stored in our microbial community, in the soil microbial community. So this is a very nice impact of the bio-based and biodegradable mulching films. So the next step of our experiment was to test these films also in field, because the results that I show you to you up to now regard some soil incubation. So soil incubation that was carried out in laboratory with a good amount of moisture in the soil, incubated at temperature of generally of 20-25 degrees. So what happened when we used these mulching films really in a field experiment? So we did a nice experiment in another part here in Italy, and in particular at the University of Reggio Calabria. We performed a mesocosm experiment, and we, as you can see here, we used these mesocosms that were buried into the soil, and we covered the soil with our mulching films. Then we cultivated, in this case, lettuce, and we tested the growth of the lettuce, and as well as also the decomposition of our mulching films. So as you can see here on the right side of the screen, at the bottom of the screen, you can see how our films start to degrade after the cultivation of the lettuce. This happen, if I well remember, after 50 days of our experiment. So this confirmed that our films are really biodegradable, but also confirm also that we need to improve. We need to improve our mulching films to allow the cultivation of more crops, because they start to decrease only after 50 days. So, going to the conclusion of our research was that we developed novel bio-based and biodegradable mulching films, and we enriched these films with nitrogen and phosphorus. We used monoammonium phosphate that allow the release of nitrogen and phosphorus into the soil, and these can act as a fertilizer, but also can stimulate the degradation of our mulching films. We enhanced soil fertility because we increased the nitrogen and the phosphorus availability. We also found that the soil type can modulate the positive effect of our films on soil microbial community, on soil microbial biomass and activity, and that the enrichment with monoammonium phosphate allow the assimilation of carbon into soil microbial biomass, so stimulate the growth of soil microbial biomass, but also a very small amount is mineralized. So these films are, as I said before, are very promising for a sustainable agriculture, because they represent a nice alternative to the conventional plastic films. and support the transition towards a more ecological agricultural practice. So the project was ended just a few months ago, but we are performing other research, other experiment about these films, because as I said before, we need to optimize the mechanical properties of our films. We need more field validation under different pedoclimatic environment, and we also have to evaluate this film for life cycle assessment and life cycle costing. I don't know. I think that I remain in time and left some time for question. But before the question, if I would like to thank all the people that were involved in this nice project, in this nice research that we carried out during the last 2-3 years here in Palermo, in collaboration, as I said before, with the University of Hohenheim, with the Professor Ellen Candler, with other colleagues from University of Reggio Calabria, from University of Ferrara, and also I like to thank the team, the research team here at the University of Palermo, and all the people that were involved. And a very nice aspect, beside to increase soil microbial biomass, increase soil fertility, and so on. The nice things of this project was the relationship that we experienced, that we got with the people, also sharing a nice cup of beer and pizza in different part of the unit research involved in this project. So thank you for your attention, and I hope that there are a lot of questions or curiosity.
So thank you, Armando, for your very interesting and inspiring presentation. And now I open the floor for the question. So if there are questions, please feel free to raise your hand. Okay, so we have one question from Fernando.
Congratulations for your presentation. It was very interesting. I think the topic it's very appealing, considering how the mulching can impact the microbiome. Is the same principle of cover crops as well. I have done some work on mulching using plastic with different colors, because we know that we have different thermal and radiation properties, and this is going to impact on the growth of the plants as well. My question is, have you considered different colors for your mulching? Because that's going to have some implications on the development of the plants.
Oh, okay. Thanks for the question. I missed the last slide, so I share again my presentation. So this is our films. This is the films that we prepared. So you can see here they are almost black. We used the different colors. We got white or transparent films, the black one, and also the green one, because what we see, yes, we tested the different colors, but we tested just not in field, not in laboratory, in soil incubation, but only if we were able, using this component, cellulose, chitosan, and so on, to obtain different colors. This is a very nice aspect of the mulching films, because as you said before, different colors may impact on at least on the soil temperature, And also another important aspect of the mulching film is, yes, is soil temperature, but also the visual impact that the people have in seeing a lot of hectares covered by plastic films. So we tested the white or the transparent films, we tested the black one, and we also tested the brown and the green one. But in field, we tested only the white one, and now we are starting to test the black one. This aspect, we got these films, the black one, by using pulverized biochar. So we used not again not the fossil based sources, but again bio based sources. We pulverized the biochar. And we mixed the biochar with our dispersion, and what we got was this dark gray, not black, dark gray films.
Okay, thank you very much. As you mentioned, the effect on the temperature, it's something significant, and in your case, we found that the red color was more suitable for tomatoes. Yeah, thank you.
The red color, no, I'm interested in this in this aspect. So the red color was performed well, better for tomato.
Yes, we tested blue, red, gray, black, and untreated, uncovered. Okay, and
Okay.
the most suitable was red.
For tomato.
Yeah.
Good. Nice.
Thank
Okay,
you.
thank you. Are there other questions for Professor Laudicina? Maybe I can ask. Can I ask one?
Please.
Okay, so maybe I lost this during your presentation, but I mean, I understood that the idea of these, let's say, bio-based, biodegradable mulching plastics is that at the end of the, let's say, of the cultivation season, you can basically bury them to improve the, let's say, the characteristics of the soil. But my question now is, if this, let's say, burying of the mulching plastics occurs at the end of the cultivation season, what is the rationale of having, let's say, nutrients like nitrate and phosphate, which can be, let's say, released when, let's say, the production is over? Because I mean, while I understand that it is important, like to improve the carbon content of the soil, the microbial biomass, but if you release nitrate, which is very mobile in the soil, and you release phosphate, which is on the other hand subjected to immobilization process, what is the rationale of having these nutrients?
Okay. Thanks, Yuri, for the nice and very interesting question. So, as I said before, the idea was to add these films to release nitrogen and phosphorus, and also to improve nitrogen and phosphorus availability. Right, to add this element at the end of the cropping cycle cannot be so useful. You are perfectly right. But as you know, this is reasonably for nitrate, because nitrate is not retained by our soil. Generally, it is very easily removed. But for phosphorus, this could be very important, because phosphorus released by our films can remain in the soil and can be useful for the next crop. But the main aspect, I think, about these films is not also, but is not mainly the supply of phosphorus and nitrogen as a fertilizer. But what I said during the presentation, we modified the carbon to nitrogen ratio, the carbon to phosphorus ratio, and we experienced at the end a ratio that was more useful for soil microorganisms to decompose at the end of the cropping cycle these films. So anything remained in our soil, and the plastic was completely decomposed. Okay, then about phosphorus, as I said before, phosphorus can be retained by our soils. About nitrate, it is very easily leached out from the root the root zone, but this depend also on how we manage irrigation. We are in a semiarid environment. We do not have a lot of water, so if we are very if we use the water in a right way, we can retain also these forms of nitrogen in our soil. Anyway, many thanks for the nice question. I will speak about this with our PhD students.
Thank you for the answer. Now we have a question from Cristina Micheloni. Please.
Yeah, hello. Thanks a lot. Thanks a lot for your presentation. Very interesting. And my question is somehow related to what was just asked, because I was puzzled a little bit how to fit it within the crop rotation if we are talking about annuals, or within the cropping system if we are talking about perennials. And so I was just imagining that the degradation and the peak of availability of nutrients is about 4 months after barring, if I am correct, 3 or 4 months, something like that. And so I was imagining that this degradation and following availability of nutrients is also a function of the temperature, the environmental temperature. So if we imagine a cropping system for vegetables, probably you bury it in autumn, that means November, potentially, in Sicily, maybe here in Friuli a bit earlier. But then it means 4 months later, you have another crop. So we can imagine it in a crop rotation.
Yes, sure. But
sorry, just I complete the question. But it depends also if this availability is not negatively impacted or somehow modulated by the temperature. Because if I imagine in November, December, January, the temperatures are lower, maybe the availability of nutrients a bit later. That anyhow, it could be fitting the scope anyhow. That was my reasoning. Different is in perennials. I was imagining vineyards or orchards, and then I don't know how to fit it. Then I am a bit lost. But my original question, I put the two of them together, was if there is a reason why you decided to use chitosan from sea animals and not from fungi production, as actually for cost reason, basically the majority is focusing on fungi production or microbiological production, let's say.
Okay, thanks for the question.
Thanks a lot.
Also nice because allows me to explain why we selected chitosan first of all. So we think in chitosan because our first idea was to use some components that hold nitrogen, and as you know, chitosan hold nitrogen. So our first idea was this: use cellulose and a small amount of chitosan because we have nitrogen. Then another important aspect: we do not use cellulose or chitosan or sodium alginate from biomass, from crustaceous, from algae. We bought this component. Okay, what I said before was that this component can be also obtained from different sources. But for the first test that we did, in our laboratory was to test the, as I can say, the product from Sigma Aldrich or other company, just to have the pure products and test these. Then you are right, we can use chitosan from fungi, chitosan from insect, because if we think also to the Asian market, we have a lot of chitosan. And so chitosan can come from different sources, but the bad drawbacks of using chitosan, we experienced some problems with chitosan. As I said at the end of my presentation, we need to test these films also for the life cycle assessment. At this moment, we get a very high cost. They are not so they do not have advantages from economic and environmental point of view. So we are thinking to replace chitosan with other components. This also because chitosan allow the films to be more rigid and not so plastic. So we are thinking to replace chitosan with other sources. And we do not need to use chitosan because really we can add nitrogen and phosphorus by adding inorganic salts. So we can change chitosan and replace these maybe with again with other components. We are working on this. But your question is perfectly right. And the other aspect about your question, we found the release of nitrate that started the peak. We reached the peak after 30 days, whereas about phosphorus about after 60 days. And they completely decompose after 120 days.
Okay, thank you, Armando. We have a question in the chat by Gianni Trioli. He asked, very nice topic. Have you done an estimation of the cost per surface unit of the biopolymer you develop?
Okay, so the cost, we did also this evaluation. The cost is slightly 20 from 15 to 20% higher than the commercial available mulching films that are available as bio-based. I do not want to mention the name of the company. Okay, so our films are have a higher cost, but if we think about the cost that the farmers have to as I can say, the cost about the removal of the plastic films, if we also take into account this, at the end, we calculated that by using our films, the farmers can save. If I well remember, it was we did this in a business plan for the startup based on this film. If I well remember, it is about 150 to 200 euros per hectares. So farmers at the end can they will our films again have a higher cost, but at the end the farmers get some income because they do not have to remove this plastic from the soil.
So they spend more at the beginning, but they can save money.
Yes, can
in
save
the long
money
run.
at the end. Yes.
Yeah, and that could be a good opportunity. So other question, I saw a hand raised, but then it disappeared. I couldn't read the name of the. Okay, so Maria, what do you say? We don't have any more questions.
Thank you so much. Yeah, if we don't have any more questions, we can close. We thank very much Professor Laudicina for this first webinar of the second series. And
you're welcome, and many thanks again for inviting me. Many thanks.
Thank you again, Armando. And I would like just to remind that the next appointment for the second webinar series of the CEDRIC project is in more or less 2 weeks, on the 23rd, if I remember correctly.
5th, I think.
25th of February. Yeah, so the next appointment is in 2 weeks. And again, thank you, Armando, and congratulations on your very interesting work.
Yes, really compliment for your research.
Thanks. Bye. Again, thanks to everybody.
Bye.
Bye. Thank you.
Thank you.
Bye.